everybody, this is Mrs. Kraus. Real talk, this is like the fifth time I've tried to record this video, so it may not be the highest quality, but this video is for the Unit 7 Lesson 3 notes, which we started in class, but I'm still going to go over the whole thing with you, and you can access the notes packet if you don't have it on paper by going to Unit 7 Lesson 3 on Blackboard, and you can download the Word document file. I'm going to have to make several videos, not just one, because YouTube limits you to 15 minutes per video. Okay, so let's get started. Alrighty, so in lesson 1 of this unit, we learned about experiments that scientists did to confirm that DNA was the genetic molecule in viruses and living cells. One experiment was done by Hershey and Chase to show that viruses injected their DNA into a host cell. So that showed that DNA was probably the genetic molecule in viruses instead of the protein coat that surrounds the DNA. Avery and McCarty and Macleod did an experiment to expand on Griffith's pneumonia bacteria experiment, and they showed that DNA was being passed from dead S strain pneumonia bacteria to living R strain pneumonia bacteria to make the R strain S strain, so to make them harmful. And so they figured out that DNA was the genetic molecule of the pneumonia bacteria because it was transforming this non-harmful bacteria into harmful bacteria. So if DNA is the genetic molecule in cells, then what are proteins used for? They actually do a lot of different jobs in the cell, and that's why they're called the workhorses of the cell. Here are 5 possible functions for proteins. Antibodies are proteins in your immune system that will mark pathogens like bacteria for destruction by your white blood cells. You have keratin protein in your hair and nails that's used for structure. You have enzymes that are used for speeding up reactions. We talked about 2 examples of enzymes that assist with DNA replication in our last notes, and those 2 enzymes were helicase and DNA polymerase. You also have proteins like hemoglobin in your red blood cells that are used to transport oxygen. And then you have proteins that are used for muscle movement. We're going to skip the next slide, just showing the structure of some of these proteins. In order to make proteins, we have to use the instructions found in DNA. Now, short sections of DNA that code for proteins are called genes, and they code for the creation of a protein polymer, which is called a polypeptide. Polypeptides are chains of protein monomers, which are called amino acids. Now, we do have an issue. DNA is found in the nucleus, but we make proteins out in the ribosomes, which are either floating in the cytoplasm or stuck on the rough ER right here. So we need to get the message from the DNA, which can't leave the nucleus, out to the ribosome. So we have a special molecule called RNA, and it's going to take the message from the nucleus out to the cytoplasm so that a particular protein can be made. RNA is smaller than DNA, so it can fit through the little holes in the nuclear membrane called nuclear pores and travel out to the ribosome. Now, if we're comparing DNA to RNA, DNA is the master plan of all genetic information in the cell, whereas RNA is a copy of part of the master plan. What was I going to say? It's a copy of just a single gene on the DNA. Okay, some structural differences between DNA and RNA. DNA has 2 chains of nucleotides twisted around each other, so we often call it a double-stranded molecule, whereas RNA is 1 chain of nucleotides, so we call it a single-stranded molecule. In terms of the nitrogen bases that are found in each molecule, they have 3 of the same bases. Both DNA and RNA have adenine, guanine, and cytosine, but DNA has thymine as one of its pyrimidine bases, whereas RNA has a new base called uracil. Also, their pentose sugars are different. DNA has the sugar deoxyribose, RNA has the sugar ribose, and they really only have 1 difference in their structures. Deoxyribose is missing a single oxygen atom here. Now this is a short mRNA molecule, and notice that it has the base uracil instead of thymine. Now, since uracil is filling the same role in RNA as thymine does in DNA, if we were asked what base does uracil pair with, it's going to pair with adenine since it's doing thymine's job. We've got 3 types of RNA used in protein synthesis. Messenger RNA or mRNA copies DNA's code and will take it out to the ribosome. tRNA or transfer RNA is going to bring the amino acids or building blocks for making a protein out to the ribosomes so we can join them in a chain to make a particular protein. And then ribosomal RNA or rRNA is a component of ribosomes. Ribosomes are unique organelles because they're not made out of membrane like most other organelles. Instead, they're just made of rRNA and proteins. So this is a picture of mRNA, just a long straight chain of nucleotides. Again, this is what leaves the nucleus and travels out to the ribosome. This is tRNA. It has a unique shape. It looks like a 3-leaf clover, or actually it kind of looks like a T too. And each tRNA has a spot where an amino acid or a building block for making a protein will attach. And there are 20 different amino acids that the tRNAs can carry. This is ribosomal RNA or rRNA. It's doing its little dance. And again, this is just a part of the ribosome along with proteins. All right, so protein synthesis happens in 2 steps, transcription and translation. During transcription, mRNA is going to be made to copy the code from a single gene in DNA and take it out of the nucleus to the ribosomes. DNA can't leave the nucleus because it's too big, but we also want to protect it since it's the master plan of all genetic information in the cell. All right, so first we need our DNA to unzip, just like it does in DNA replication. And then RNA polymerase, which is a new enzyme, is going to pull in free RNA nucleotides and match them up with nucleotides on one of the parent DNA strands to create an RNA chain. Once we've made our mRNA chain, it is released and it leaves the nucleus through the nuclear pore. This is similar to replication because the DNA has to unzip and because an enzyme is used to pull in free nucleotides to build a chain. So you have this picture in your packet. RNA polymerase is this light blue bean-shaped enzyme in the background here. It has 2 jobs. It's unzipping the DNA just like helicase does in DNA replication. And this is our DNA here in blue. And it's pulling in free nucleotides like the one shown here to match up with complementary bases on the parent DNA. So we are building this mRNA molecule in red. Now, if we were transcribing an mRNA sequence from this DNA, remember, mRNA does not have thymine, so where we would put a thymine, we're going to put a U for uracil. So I'm just going to fill in the complementary bases here. And this is the sequence that you should have. Okay, delete that so it doesn't stay in my presentation. Okay. Now, part 2 of protein synthesis is translation. During translation, tRNA is going to read the instructions in the mRNA molecule, and it's going to join amino acids together to create a polypeptide. This occurs in several steps. The mRNA has to travel to the ribosome. Then the tRNA is going to bring the amino acids to the ribosome and join them in the order specified by the mRNA instructions. Those amino acids join together by peptide bonds, which is a special type of covalent bond, and it makes a polypeptide chain. Now remember, proteins are usually multiple polypeptides folded around each other. For example, hemoglobin has 4 polypeptides that are folded around each other. Now, how does tRNA actually read the mRNA instructions? Well, if you see We have 3 bases on our mRNA molecule down here called a codon, and we have 3 complementary bases at the bottom of this tRNA called an anticodon. If the anticodon on a tRNA molecule matches with the codon on the mRNA, the amino acid that the tRNA is carrying up here gets added to the chain. And here we also see the bottom of a tRNA matching its anticodon to the codon on mRNA. So this is a picture that you're labeling in your notes. We've got the ribosome, which is in green in the background. We've got our mRNA molecule here, which is read in sequences of 3 bases called codons. These blue tRNAs match their anticodons with the codons on mRNA and add their amino acids to the chain. Okay, this is just a little flip book where we can see translation in action. We've got our blue ribosome and our mRNA here. Notice we've got a tRNA coming in, matching up its anticodon with the codon on mRNA, and it's going to add its amino acid to the chain. Now we've got a second tRNA coming in. Those amino acids will join together by a peptide bond, and then we're going to kick out our first tRNA, and it leaves its amino acid behind as it adds it to the growing polypeptide. This process continues until we reach something called a stop codon. And this sequence in mRNA doesn't match with any tRNA anticodon. So at this point, the polypeptide is done. Okay. Now you have this video that's linked in the presentation. I think it's really helpful because it shows everything in action. Okay. You're also going to be making this flow chart. It shows that the process of converting the information in DNA to make an mRNA molecule is transcription. And then tRNA is going to translate the information in mRNA to a polypeptide or protein. That's happening out in the ribosome. And that step is called translation. Okay, so this image here shows us both transcription and translation. I'm going to kind of walk you through it. We've got our big circle for the cell and 2 smaller circles for the nucleus and ribosome. The ribosome is actually much smaller than the nucleus, but we want to be able to see what's going on inside of it. So here we've got our double-stranded DNA being used to make our single-stranded mRNA. That process is called transcription. mRNA leaves the nucleus and travels out to the ribosome. Okay. In the ribosome, our little green tRNAs are going to use their anticodons to match up with the codons on mRNA, and then they'll add their little purple amino acids to the growing polypeptide chain. And this step that's happening in the ribosome is called translation. You can think of making a protein like baking a cake. This is real old school, but if you had a recipe book at the library, that might be like your DNA since it's got all the genetic information in the cell. The nucleus would be like the library. You're just going to make a copy of one recipe, so that would be your mRNA, like making a copy of one gene, and you can take that out of the library, and you, the tRNA or the baker, you're going to read that mRNA recipe, and you're going to put the ingredients together. The ingredients are like your amino acids, and you're doing this in the ribosome or the kitchen. And if you put your ingredients together in the right order, you make your polypeptide or cake. Okay. I'm going to stop the video here and I'll make a new one in a minute.